how they get interconnected when we talk about ecosystem. So human, animal, and when we talk about the environment, they are connected with each other at multiple aspects to make the entire ecosystem work. And antibiotic resistance is one of the important criteria which exemplifies this interdependence. So we look up to different infections and different microbes like Salmonella, Campylobacter, E. coli, which keep on circulating between different aspects of these parameters which are concerned with the environment. So if we talk about humans, livestock, and the environment itself. So when they are moving from one to another, they are also trying to evolve. And in the process of evolution, they do acquire some resistant genes, which make them, more sustained in different diverse environment. And as we know, it's a survival of the fittest. So those changes are being acquired, which help them in surviving extreme environments and diverse environments as well. And how they acquired it? They acquired it through different ways. There could be some kind of genetic means like plasmids, integrons, and transmons. So these are nothing but genetic segments. So when I say they acquired resistance, so what do I mean by resistance? There will be a gene which codes for some protein, and those proteins give up resistant to certain antibiotics, and then can be in different forms, those genetic elements like plasmid, integrons, or transposons. When we do metagenomic studies, it has been seen that wastewater resistomes overlap with clinical isolates. So there is somewhere a great correlation between what infections we see in clinical setups and what we try to isolate from wastewater fields. They are showing some similar strains, and that is very surprising because somewhere these strains are being passing from one place to another. So WHO reports, if we look into that antimicrobial resistant causes around 1.27 million deaths annually worldwide, with majorly coming from the foodborne pathogens contributing to significant responses. So this says that when we talk about food, when we talk about poultry, livestock, we need to make sure that the influence of antimicrobial resistance is also playing an important role. So if further we go into the deep down studies, analysis show that there are 36,822 different human salmonella isolates in China, which were seen in the year 2014 to 21, and which were found to be 30% resistance to these antibiotics, highlighting cross-sectoral transmission. Same way, we have fluoroquinolone-resistant Campylobacter strains, which are widespread in poultry, and transmission to humans occurs via contaminated meat, causing gastroenteritis that is harder to treat. So, these strains, when we say that they are coming from livestock or when they are coming from poultry, but it depends on how we are handling it. So cooking it properly is one of the way. How you process is what hygiene conditions you are maintaining. And second thing is sometimes the bacterias which they are, because these poultry and livestock are on high antibiotics. So what happens? They are having now those microbes which were antibiotic resistance, and so they become much more harder to treat or to get rid of. Another strain we see when we talk about is vancomycin resistant enterococci, detected in both hospital patients and from animals, spread through basically manure application in agriculture, contaminating water and soil samples. So it's not only if we deal with the livestock, we are coming in this picture, but we should understand that livestock and poultry are a part of the ecosystem. Okay, so through different means, they are also allowing the breach between the soil and what they have in their body. So when they when they are, moving around in the soil, in the ecosystem, in the environment, they are also releasing these microbes outside. And these microbes are then contained in, deep down into the soils, further affecting our crops, the land fertility, quality, and sometimes it also seeps into the water which is nearby, because through rainwater, those upper layer of the soils get washed away, and those layers do contain these antibiotic resistant strains. So one again major thing which we see is the carbapenem resistant Klebsiella that has been found in rivers near urban centers, that highlighting environmental reservoirs. Again, that's what I was saying. Ki, from livestock, poultry, they are coming down to the level of soil where they are damaging the crops, and now from the soil they can also mitigate into the nearby water bodies, where again the water also gets contaminated with the resistant strains. So when we say antibiotic flask, what do I mean? I mean basically the dynamic movement of antibiotics and resistant determinants across different parameters of the ecosystem. So who all are there in this parameters of ecosystem? They are nothing but the humans, the animals, and the environmental microbiome. This together makes up the ecosystem. So pathways of flask could be clinical use like hospital effluents. So, as already seen, resistant Klebsiella species, E. coli species are frequently detected in sewage. Veterinary use, they come down to the level of manure and then soil and finally reaches to the crops. And then we have ESBL producing E. coli, which is a resistant strain traced from poultry farms to vegetables. So, something which is going into every aspect of the ecosystem and disturbing the quality. Aquacultures, water system, wild species, the fluoroquinolone resistant aeromonas found in fish and rivers. So now the fish are having those depositions of this microbes with them, which are highly resistant. The rivers are also getting contaminated with them. So when we say there is a resistant, first of all, from a microbiome perspective, what is it actually? So as I already mentioned, that there is DNA. DNA is basically the information which gives us what protein should be produced. So now there are different, like if I want to target a microbe, there are different aspects which can be targeted. One is you can inhibit the growth of the microbe. Now, when I say that inhibition of growth has to happen, that can also be done at different levels. The first level could be destroy the machinery which is used to produce proteins. If the microbes are unable to produce protein, they cannot have the resistance, right? So, inhibition of protein synthesis. The next could be don't allow them to multiply. So, if for multiplication, one important thing is replication. You should have your genetic material duplicated, then only it can be passed on to two newly formed cells. So it's better you inhibit the process of DNA replication or nucleic acid synthesis in simple words, so that further, amplification of the number of cells will be down, or inhibit the cell wall synthesis. If the cell wall is not there, they will not be protected from the environment. They will be destroyed. There will be lysis of the cell. It can also disrupt the function of the cell membrane, which helps it in preventing and creating a boundary with the environment. Other than that, we can also have different pathways which can inhibit the different aspects of metabolism in the microbe. So these are the different ways which the antibiotics actually work. So when we say antibiotic and when we say resistance, so initially an antibiotic is designed to kill a bacteria. It could be by either of those means which I talked about, where it inhibits protein synthesis or it inhibits the genetic material synthesis or it damages the cell wall or something else. But when a resistant strain is there, what happens in that process which was inhibiting or which was destroying a particular important cellular pathway is now no longer working out. So, one is enzymatic degradation, that is by beta lactamase. So, bacteria they produce the enzyme that is called uh, this extended spectrum beta lactamase that hydrolyzes antibiotic molecules. So, now you have given an antibiotic, but the bacteria is not getting killed. Something is there that because of which is not getting killed. So what is it? So they have an enzyme which is degrading that antibiotic. So now no longer that antibiotic can work on the bacteria and degrade the bacteria. So they become resistant and they can survive even if the antibiotic is given. So two things happen over here, that you are keep on giving antibiotic thinking that sometime it will work upon. Second is there is a saturation which has reached. Now the bacteria has evolved thinking that no, this is the normal condition, and if they want to survive, they need to adapt to this environment. So they have become such a strain that they even there is a presence of antibiotics, they are not going to be killed. So ESBL producing strains are mostly seen in hospitals, and they are also reported to be there in some outbreaks that limit treatment options, because if we know that a particular bacteria is susceptible to a particular antibiotic, we will do the treatment accordingly. But what if the drug does not work? Then it becomes difficult to do the treatment. So there comes a challenge where we need to figure out other ways of treating it. Flux pumps, membrane proteins actively expel antibiotics from bacterial cells like Pseudomonas aeruginosa. Uses this particular efflux, which is MEX AP OP-M. This is a bit complicated, but it's actually the name of the pump. What it does, it resists fluoroquinones. So now those fluoroquinolones were supposed to enter, but this pump is not allowing it to enter. It is putting it out back, causing resistance. So it causes broad spectrum resistance. One pump can expel multiple drug classes outside, and hence drug is not able to reach the target. And if the drug does not reach the target, again there is challenge in treatment. Other thing is target modification. So antibiotic binding sites reduces drug efficacy. MRSA modifies like multiple resistant strain, which is there. It modifies the penicillin binding protein, making beta lactams ineffective. Mutations in the gyrase confirm fluoroquinol resistance in the Campylobacter. So nothing to worry. From different perspectives, I'm just trying to convey that there are different strategies during the time bacteria has evolved to get rid of those antibiotics. Biofilm formation, bacteria embedded in extracellular polymeric substances, reduced antibiotic penetration, staphylococcus. So, when there is a mix of bacteria, a consortia which makes a film, they kind of secrete a slimy layer on it. And that layer is actually not allowing certain drugs or certain antibiotics to cross and reach the bacteria. These kinds of biofilm you can see on catheters, which are used in hospitals. You can also see on some uh, surfaces which are very close to water bodies or which are very close to soil surfaces. They can have it. And it can lead to chronic infection in some cases, like cystic fibrosis, lung infections with Pseudomonas. So, again, talking into the aspect that they are very important when we say that they are generating resistant strains. So, when we say how they are acquiring it, they're acquiring it through a technique which we say as the horizontal gene transfer. So vertical is from one generation to next, but horizontal is basically in the same generation. So here I can give an example where bacteria A has a resistant to uh, suppose antibiotic A, whereas bacteria B has a resistant to antibiotic B. Now, if they come in combination, they exchange genetic material among themselves by a horizontal gene transfer. And now, because of this, bacteria A can have resistance to both antibiotic A and antibiotic B. So, what it does, it now gives or generates a multi drug resistant strain. So, what are the different ways it can do so? Conjugation, transformation, and transduction. So, conjugation, for this, we need two molecules in place. One should be the donor and one should be the recipient, like two bacterial cells should be there. And they are exchanging genetic material among themselves. Next is transformation, where we don't need a donor bacteria, we only need the bacterial cell. And with different constructs from different environmental sources, they can get an extra genetic material inside, that is your plasmids. That the process is called transformation. How it is different from conjugation? Conjugation, we should remember that it is between two bacterial cells, where one is donating and another one is accepting it. But in transformation, there is no donor bacteria. It is only acceptor bacteria, and it is taking up the genetic material from other sources other than the bacterial source. Other than that, we have a transduction process where we use bacteriophages, which try to embed the genetic material inside the bacterial cell. So you can already see the pictorial presentation of it, where a bacteriophage is coming and sitting on the bacterial cell wall and trying to release its genetic material inside the bacterial cell. So when it is releasing, that genetic material can give it some resistance to other antibiotics. And as a result, this plays a very important role in generating strains which are highly resistant to antibiotics. Now, when we say all these, there are three important things which are getting influenced by the resistance. One is human health. So, overuse and misuse of antibiotics. WHO estimates that up to 50% of antibiotics are prescribed inappropriately in human medicines. In India, studies show high rates of fluoroquinolone resistance in E. coli linked to overprescription for diarrheal diseases, accelerates resistance in community acquired infections. So that's what, as of now, we know COVID can be spread from one person to another. What if we generate a bacteria because of all this um, excess use of antibiotics again, which gets transmitted from one person to another, which may be now a simple bacteria like E. coli, but that can get mutated and acquire resistance and can cross barriers. It can even go worse when we talk about livestock. The second important part when we talk about human health is the hospitals, which are releasing this effluents as reservoirs of antibiotic resistant strains. So wastewater, which contains high concentration of resistant bacteria and antibiotic residues. Carbamazepine resistant Klebsiella pneumonia detected in hospital effluents. It's a case study where it's shown in Delhi, later found also in the nearby river systems, because they all are connected. If it is getting sourced at one particular point of a water body, it will get communicated to the other point, and from there it will also get communicated to the land. Hospitals act as a hotspots for resistance gene dissemination into the environment. So not only it is restricted to hospital, it goes to a water body and can sip into a different water channel finally and go to a different place where the soil again get mitigated from the water body. So ultimately it is coming in the ecosystem. Now that was from the human perspective if we talk about. The next one is animal health. Growth promoters in livestock. So antibiotic use subtherapeutically in poultry and cattle to enhance growth. ESBL producing E. coli in Indian poultry farms showed greater than 60% resistance to these kind of antibiotics and more than 40% resistance to ciprofloxacin. So what it shows that there are increased, you know, strains which are resistant, and they spread to humans via the food chain. Zoonotic transmission is the next part where we see that this, different species like Salmonella, Campylobacter from poultry are known to cause the major foodborne diseases. Chloroquinolone resistant Campylobacter strains in poultry linked to human may cause gastroenteritis, and these outbreaks are already reported in Europe. There is a direct evidence of resistance moving from animals to humans. So when we say humans, when we say animals, and when we say ecosystem, we cannot separate them. They are going to be linked at multiple aspects. So there is a one scenario where we have talked about the zoonotic diseases outbreaks in India. So in India, there has been seen to have an increased incidence of zoonotics outbreaks from 8% between 2018 and 23, with Northeast having right now 35.8% and southern regions having 31.7%. So this shows what how important it is to look into the animal health also. Only human health may not be the solution to everything. The next, when we say go ahead, is about the environment, because this is a place where we all are, getting connected. So wastewater, soil, and aquaculture, antibiotic residues and resistant bacteria, they are going to enter the rivers, lakes, the soil through effluents and manure. Studies in China has shown that there is incidence of multi-drug resistant E. coli strains, Aeromonas species strain in aquaculture ponds, which are linked to fluoroquinolone use in fish. Soil fertilized with livestock manure carries resistant enterococcus strains and E. coli contaminating the crops. So what? That's what. Again, it's comes to the soil and then it reaches your crops, and again it is going on the plate. So this is where the antibiotic, which is being coming from one source, now mitigating through 10 different places, and again coming back to the humans in the form of the crops which they are using for their day-to-day food purpose. Antibiotic residues altering microbial ecology. In low concentrations of antibiotics in water select for resistant microbes. Wastewater treatment plants in Europe selected resistant strains persisting despite treatment. Environmental resistomes overlap with microbial isolate, showing continuous flux of them in the environment. Now, when we say there are lots of issues with all these things, that doesn't mean that they are still going on. There is a lot of work also which is going on to take care of these things. So, at the global level, we are having different organizations who are looking into it. The first one we talk about is the WHO GLASS, which is the Global Antimicrobial Resistance Surveillance System. It is established in the year 2015 to standardize AMR data collection worldwide. It provides global stats on resistant trends in pathogens like E. coli, Klebsiella, Staphylococcus aureus. GLASS 2024 shows that more than 40% resistance to fluoroquinolones is E. coli urinary tract infections across multiple regions. That means this particular resistance is being acquired by E. coli, and it is not restricted to one area. It has spread to different locations. Now, how the spread is happening? There are different ways. The next is FAO, which is the Food and Agricultural Organization, overuses on antimicrobial use in agriculture and food production. It promotes narratives to antibiotics in livestock, such as probiotic and improved biosecurity. Supported projects are there in Southeast Asia, which are going on to reduce the use of antibiotics in poultry farms for better practices, which reduces the stress on the environment. And there is one more organization, which is WOHA, that is World Organization for Animal Health. It develops international standards for veterinary antibiotic use. It monitors AMR in pathogens like Salmonella and Campylobacter. OIE reports highlight high resistance of antibiotics in poultry associated with Campylobacter in different parts of Europe. So these are the different strategies which at the global level are concerned with regulating the usage of antibiotics. But there are different ways by which we do check the presence of microbes, and also we do check that whether this microbe is a resistant strain to a particular antibiotic or not. So the first one talks about the metagenomic studies for resistome uh, analysis. So here we take samples from different environment, we soil samples, wastewater samples, samples from gut microbiome also, to identify the presence of resistant genes, which help us figure out if this particular bacteria is now becoming resistant to one particular antibiotic, what could be our next strategy. Metagenomic studies in Indian rivers detected the presence of BLA NDM1, which is nothing but a carbon amase uh, enzyme, which now present in the downstream of hospital effluent. It captures unculturable microbes and reservoirs. So some places, some microbes, it's difficult to culture them. There we use this technique so that we can get to know that in a vast consortia of microbes, because when we say we are taking samples from different environmental sources, we know that there is not only one microbe, there is a mixture of microbe, and that mixture of microbe will have thousands of different DNAs with it. So this will help in identification of a particular gene which is more highly seen and which has the gene of interest like the resistant gene with it. So viral metagenomics is also a concern when we talk about human health. So it could be done from freshwater and marine water samples where it identifies known waterborne viral pathogens, could also look for the emerging waterborne pathogens and identify the fudge originating from specific fecal sources, remediation of fecal contamination in water. Could also help in identification of foodborne viral pathogens, identification of emerging foodborne pathogens, the fudge originating from specific samples, and the remediation of fecal contamination in food and soil. Similarly, it could be done for the wastewater, where we can have enteric and non-enteric pathogen being screened out, and then what are the different viruses which are sourced from community for surveillance purposes, and the characterization of novel viral indicators from different samples. Air can also be a source where we can use the metagenomics to isolate some specific viruses which can become pathogenic and also can become infectious. The next tool when we go ahead is talking about the culture-based diagnostics. So this is again another method which talks about the traditional culturing technique where we can have disk diffusion, MIC determination to know that what whether a particular and microbe is susceptible to a particular antibiotic. First of all, do it comes in the range which is the standardized range? If not, then what is the minimal inhibitory concentration and what is the minimal bactericidal concentration? It also provides the phenotypic data. Time consuming process it is, and it misses non culturable organisms. So some organisms like viruses and other microbes may not be cultured, and it could be difficult to examine them. When we say about culturing, we have three different ways of doing it. You can have a spread plate method where the media is present, and then you can have some culture being spread on that particular media and then incubated. So that will give you surface colonies, evenly spread it can have. Another is pour plate method where we have the culture and then we mix it with the agar and then do the plating. Again, it gives us some colonies which are on the surface and which are some which are subsurface, that is in between the agar. And the most important, which we generally use, is the streak plate method. The beauty of this method is that it allows us to get single colonies, pure colonies, because we do multiple streaks, and at the end, we come up with the lower load of the microorganism, which helps us in isolating single pure colonies. Other than that, we have laboratory tools where we talk about the molecular diagnostics. When we say molecular diagnostics, when the prime instrument which is used is PCR, real-time PCR and whole genome sequencing, which helps in the rapid detection of resistant genes. The PCR assays for black XM and drug-resistant genes like MCR1 in livestock samples can be done. It is very like advantageous compared to the culturing techniques, in that there was challenge that some microbes are not culturable. So in those case, we can go for PCR. Second thing, it does not take that much time which is taken for culturing. It is less time taking and it is more specific in nature. So just the basics of PCR, you have few components in it, you have to mix them and then allow it to be settled on a thermocycler. After that, after few cycles, so you have different steps for each cycle. In one cycle, you have first denaturation, where the initial genetic material should be separated from each other. Then we anneal it with specific primers which help in amplification of that. So it's basically like if we have one dot in a white background, it is very difficult to identify that dot. But if we have 10 dots or 100 dots instead of one dot, then it is easy for identification. So that's what she says, that PCR helps in amplification of those genes, which increases the visibility to figure out the presence of that antibiotic resistant gene in the microbial communities. So then we come across the different types of interventions which can happen. So it could be two different ways out where we talk about antibiotic stewardships. So rational prescribing in hospitals and clinics, there should not be unnecessary uptake of antibiotics in different places, even in veterinary shops. Stewardships programs in US hospitals are there, which reduces the broad spectrum antibiotic use by 20%. Further, it's shown to reduce or lower the rate of clostridioids difficile infections. So that says it's a case study which is showing us that yes, if we reduce the usage, we are trying to cut the spread of the infection. That means the particular strain becomes more resistant to the drug, because see, we should understand, as currently the climate, the temperature in summers go around 42, 45. It was not that this temperature we are not able to survive. We are surviving. The same way, when it becomes same thing for bacteria, if they are continuously in presence with those antibiotics, they think that it's a normal thing for them. They don't think it is a stress condition, and they get evolved accordingly. Veterinary stewardship also bans on growth promoters in EUV. So they have restricted the use of using these growth promoters in Europe since 2006, and it has shown to be really effective. Alternatives to antibiotics. So if we are restricting our use on antibiotic, what should be the like replacement? Go for more of probiotics, which is good microbes, competitive exclusion of pathogens in poultry gut, fudge therapy, targeted killing of resistant bacteria. Case trials are going on in the places like Georgia and Poland, which show promise against some of the resistant strains like Pseudomonas infections. Usage of green chemistry, development of biodegradable antimicrobials that reduce their persistence in the environment. Though we are using antibiotics, antimicrobials, if they are going down in the environment, they are still there and they are hampering the ecosystem. So we should look for alternatives which, if they go in a particular environment, they themselves get degraded. Their nature should not be supported by that. That can be done with the help of green chemistry. So moving ahead, we all came across that the importance of One Health and all the three parameters is important to work in function and move together. So surveillance strategies for zoonotic disinfection regulation is again important, where we say that wildlife monitoring should be there. Preventing zoonotic diseases outbreaks requires a multifaceted approach to surveillance, wildlife monitoring, because this also reduces the effect. Okay. Now, coming to the key takeaways, where we say One Health link humans, animals, and environment, making antibiotic resistance a shared global challenge. Bacteria resist antibiotics through enzymatic degradation. We have seen the different ways they pump out the entire antibiotic and they survive in those conditions. They modify the target so that no longer it can act on the target, and as a result, the drug become ineffective. Formation of biofilms, which again does not allow the drug to move inside, and horizontal gene transfer, where they get resistance through the acquiring genetic materials like plasmids and all, which are carrying the resistance to the particular antibiotics. So antibiotic flux occurs via clinical misuse, livestock practices. That's why it's important over here when we talk about veterinary, livestock, agricultural practices, and environmental reservoirs like wastewater and aquacultures. Global surveillance strategies, which are taken by WHO, FAO, and OIE, and advanced tools can be helping in tracking the spread of resistance, because these global surveillance studies gives us reports, gives us the prevalence of a particular strain in a particular area, because we know that area wise there is difference in these strains. The acquisitions stewardship programs can help, and innovative alternatives like probiotics, flush therapy, and the usage of green chemistry can help us reduce the usage of antibiotics and slow the resistance cycle in the environment. Finally, giving it a culture where the environment is more healthier for the coming generations, and it is promising. So, with this, I would like to end my session. And if any questions or anything to talk about, we can go ahead.